Welcome to another episode of the ideaXme show that's profiling humans behind the really big ideas that are shaping our world, inspiring future trends and future creators, and for all those that like great stories. I'm Ira Pastor, your life sciences ambassador along for the journey today. So a few months ago on the show, we introduced the interesting topic of the importance of substrate in regenerative processes for either the maintenance of our normal tissue functions or the migration of cells, change to tissue architecture that are so very important in healing processes. And we sort of broadly define substrate as a surface or a material which cells and tissues live on, allow them to grow, obtain their nourishment, and have both biochemical as well as biomechanical function. Today, we are going to discuss some really fascinating next generation work that's going on in this domain. And we are joined today by Dr. Gulden Camci-Unal, who is assistant professor in the Department of Chemical Engineering at the Francis College of Engineering at University of Massachusetts Lowell. Dr. Camci-Unal received her Ph.D. in chemistry at Iowa State, her master's and bachelor's degrees in chemical engineering at Middle East Tech University in Turkey. And her research is at the interface of biomaterials and bioengineering, including the design, the synthesis, and characterization of a variety of functional biomaterials for applications in tissue engineering, regenerative medicine, as well as the development of in vitro disease models for personalized medicine. And she's also gotten involved in a new work in the area of low-cost point-of-care diagnostics, which we'll get into a little later on the show. And her work, interestingly, also brings up another theme that we have touched on a bit in the past, and that is that of what we'll call biomimicry, and basically looking at very interesting structures in nature, whether they be eggshells or plant leaves or marine sponges or what have you, to ultimately guide strategies that are both viable and cost-effective in bioengineering. The overarching goal of her research to improve human health, quality of life, and she's made extremely important contributions in generation of platforms for cardiac, cardiovascular, and bone tissue engineering. She has a numerous amount of research and teaching awards from the NSF Advanced Travel Award, Chevron Phillips Award, Procter & Gamble Fellowship. The list goes on and on. She has dozens of published manuscripts, conference abstracts, patents, and she is editorial board member of various publications, scientific reports, Journal of Biomaterials and Tissue Engineering, regenerative medicine research. I could go on for quite a while, but Dr. Gulden Camci-Unal, thank you for taking the time out of your schedule to come talk to us for a little bit.
I greatly appreciate the opportunity.
Absolutely. Absolutely. It's our pleasure. Typically we start off our show by handing our guests the floor just for a little bit. If you could take us into a little bit of your background story, everything from where you grew up, how you got interested in science and chemical engineering, and a little bit of your path to date in the regenerative medicine space and sort of these very interesting translational projects. I think that'll be a fascinating way to start off.
For as long as I remember, I always wanted to be in engineering and medicine fields. I'd been very much interested in different ways that engineers and doctors impact everyday life. And as a child, I also loved math and science classes the most. And later on in high school, I was very lucky that I had opportunities to see some of the different facilities that chemical engineering work on and some of the different processes that they were involved through different field trips that we had been. And this is when I knew that I wanted to become a chemical engineer. And over time, how I became interested in regenerative medicine, well, I'm a martial artist. This is a close contact sport, and I've had a number of injuries in the past. And as a patient, I was familiar with the challenges and difficulties associated with the healing process. And I started thinking there's got to be faster and better ways to improve the healing process. Then I started taking classes about biomaterials and tissue engineering and became very interested and passionate about regenerative medicine.
Outstanding, outstanding. I appreciate that background. Yeah, as mentioned in the introduction, we discussed a little bit sort of this concept of substrate previously, and it's it's always been very interesting. While the last sort of decade or two has been really dominated by sort of the stem cell centric concepts, we've seen sort of less, let's say, awareness of sort of the importance of everything else that goes on in regenerative processes, from sort of, as you've turned in some of the material, I've seen sort of this this top-down approach to thinking of things, especially in the way you write about sort of origami-inspired tissue engineering, that it's not just about putting these different things together, but they work in a certain fashion. At the same time, a lot is required for a proper substrate. There's many things in terms of cost and various biochemical factors that have to permeate, and angiogenesis, and you can't promote scarring. Really a lot on this, as you were saying, this engineering front that takes the cells and really makes something out of them. Can you talk a little bit about sort of the background of just work in the area of substrate and understanding the challenges, why it's so important, ultimately whether we want to create a nerve or a blood vessel or recreate proper bone structure? Take us on a little bit of that background journey, if you would.
Let me give you a little bit background about my research, and let me get into more specifics. So my research is quite interdisciplinary. So I use engineering approaches to solve problems in life sciences and medicine. And the motivation for me is to design functional biomaterials for cardiovascular regeneration, bone repair, wound healing, develop disease models for personalized medicine, gain fundamental understanding in biological events, as well as developing low-cost point-of-care diagnostic platforms. And the overall the overarching goal of my research is to improve human health and quality of life and give back to the society. And tissue and organ failure are major medical problems that we all know, for which treatment options include organ transplantation, surgical repair, artificial prosthesis, and direct therapies. Transplantation is oftentimes limited by the lack of tissue donors, tissue and organ donors. And to address the challenges with the lack of tissue donors and also with the lack of commercially available products, tissue engineering approaches can be used to generate three-dimensional tissues. So tissue engineering is an interdisciplinary field that involves the use of that uses the principles from engineering, medicine, and biological sciences to develop tissues that can restore, maintain, or enhance tissue function. And although tissue engineering has been successfully used to engineer avascular organs, there are still a number of challenges that exist in making highly vascularized, off-the-shelf tissue engineered tissues and organs. And some of these barriers include the lack of a renewable cell source that's immunocompatible with the patient, the lack of biomaterials with desired mechanical, chemical, and biological properties, and the inability to generate large and vascularized tissues that can readily be integrated into the host circulatory system and mimic the architectural complexity of native tissues. So to address some of these limitations, microengineering platforms can be utilized to make scaffolds that can control the tissue formation. And in my research, we use engineered materials that includes hydrogels from micro nano scale to macro scale. Some of the microengineering approaches we use is photolithography, soft lithography, 3D bioprinting, microfluidics, and emulsions to control cellular behavior. So hydrogels are highly hydrophilic and flexible cross-link polymeric networks. And examples from daily life for hydrogels include jello, contact lenses, and ion exchange resins. And hydrogels have been used as three-dimensional biomimetic environments. They have high water content, and they provide structural support for cells. And to me, the most interesting feature of hydrogels is their tunability. So tunability in cell adhesion, mechanical properties, size and shape. So in our research, we use hydrogels and also other polymeric materials for cardiovascular regeneration, bone repair, wound healing, and also for personalized medicine applications. And more recently, we have shown the use of unconventional materials for biomedical purposes. So imagine that you wanted to grow a blood vessel or a kidney or a muscle outside the body. How would you get all these cells to stick together and form the correct three-dimensional structure? So that's one of the many challenges that we see in the tissue engineering field. And the aim here is to repair or improve tissues that are damaged due to disease, degeneration, trauma, or aging. And there is a lot of research being performed to figuring out how to grow tissues at a scale and price that can meet the needs of thousands of patients a year, right? So one of the big challenges in creating those tissues is essentially simply growing them in the laboratories, because human cells require a very specific environment to shape themselves into properly functioning tissues. And sometimes it can be quite cumbersome and expensive to develop new combinations of engineered materials and molecules that can support the creation of artificial tissues outside the human body. But as we go about our daily lives, there is a diverse range of natural and also man-made materials that are overlooked in tissue engineering. So when we want to generate a tissue, right, typically we would need a scaffold, which is a very intricate mold or frame or template that guides a tissue growth. And usually these scaffolds are synthetic, but they don't necessarily have to be, really. And some of the currently available scaffolds require complex fabrication steps and specialized hardware. And in addition, artificial scaffolds can be less flexible than the tissues that they're trying to replace, or sometimes they may not have the sufficient mechanical strength to retain their shape as the tissue grows. So, and then sometimes they may be too expensive to manufacture at a scale that the public needs. But the Mother Nature may have already provided us with, with the materials that we need to grow functioning tissues. And my lab focuses on developing functional materials for repair and regenerate tissues. And one of the strategies that we use is to take unconventional approaches and utilize materials from nature or everyday life. Why? Because we want to make engineering tissues and organs simple, accessible, and inexpensive, so that our materials can be used by people in countries that have fewer resources, as well as the ones with access to the best equipment and facilities. So in my laboratory, one of our goals is to look at items that we see or throw away every day and reimagine how they might be useful for growing three-dimensional tissues in the laboratory that can later be transplanted into people. And our most recent example in this research area is to use chicken eggshells as a reinforcement in polymeric materials to generate scaffolds to repair and regenerate bone. So eggshells might appear as just leftover waste after cooking an omelet, right? But in our lab, we repurpose eggshells to fabricate tissue templates that promote the growth of bone cells and that can help them harden faster, mineralize faster. So eggshells contain minerals that have carbonate, which is also a component that is present in the native bone. So because some of the components of the eggshells resemble bone, they can be used to make those tissue templates that can replicate the biology of those tissues. So more than 2 million bone graft procedures are being performed every year, and it's crucial that the bone repair is very important to restore a patient's functionality and self-esteem following an injury. And there are limitations with the existing bone grafts, those bone graft biomaterials, and the procedures that include risk of infection, rejection by the immune system, and of course, the limited availability of bone donors. Global waste of discarded eggshells, they typically amount to millions of tons annually from household and commercial cooking. And by repurposing eggshells, we can directly benefit the economy and environment while providing unique and sustainable solutions to unmet clinical needs. And last year, we've demonstrated that eggshell particle reinforced materials can significantly increase mineralization by the bone cells, resulting in faster healing. And what we do is in this process, we insert crushed eggshells into hydrogel matrix, hydrogel mixture that forms a miniature frame to grow bone in the laboratory. And our research demonstrated that eggshell particles not only increasing the mechanical properties of those scaffolds, but also increasing the bioactivity and helping the bone cells, increasing the ability of the bone cells to grow, mineralize, and harden. And currently, we're collaborating with clinicians to understand what are the unmet needs for patients exactly, and developing products that can address those unmet needs.
Fascinating, really fascinating, and obviously thinking of you doing your martial arts, and of course breaking bones and stuff like that along the way. It fits very nicely with
it
that.
fits all nicely, yeah. Together, yeah.
Obviously, you know, clinical translation is so important. You mentioned you're working with the people on the clinical side. Other, you know, you've written about, you know, other unconventional scaffolds in terms of sponges and leaves and things of this nature. Are there, obviously I understand the connection with the eggshells and bone, any other interesting or things that we might not think of, parallels, you know, do the marine sponges as opposed to eggshells work better for maybe muscle tissue? What are some of the other interesting parallels, let's say, that you have been learning about as you looked at some of these unconventional materials?
When we use eggshells, it wasn't the first time that we have used an unconventional biomaterials. We have also taken an origami-inspired approach and used paper to grow bone tissue before, and let me explain that. So origami is the Japanese art of paper folding, and in addition to its various artistic use, the principles of origami find different scientific uses also. For instance, in stent implants, in packaging large solar panel arrays of space satellites, and in deployment of carrier bags from folded positions. And we have taken an approach where we use folded paper to grow cells and generate tissue mimetic, tissue-like structures. And the idea is to use the paper as a template material for patients who have defects of irregular sizes and shapes. So people have been using paper for thousands of years for many different applications, but it wasn't until 10 years ago that that we started using paper as a tissue engineering scaffold for cell culture platforms. And I think that sometimes simple things are just overlooked. So paper is a very flexible material. It can be easily cut, creased, folded, and manipulated to yield and to fabricate three-dimensional freestanding constructs. And paper scaffolds are very tunable in size, shape, and configuration. And in our previous study, we've constructed bone mimetic structures using paper, using arbitrary features, using regular shapes, as well as incorporating grading structures in those scaffolds. And then we have we have used those scaffolds, seeded them with bone cells, and cultured them for a desired period of time, and then evaluated for deposition of minerals by the bone cells. And we saw that the bone cells mineralize the paper templates in whatever shape that we've placed them in the scaffolds. So we also have used multi-layered scaffolds and developed a new cell culture platform to study cancer metastasis. So we also utilize paper in another area as a personalized medicine platform. So here the idea is to generate a tumor-like tissue and study how different types of cells in the tumor interact with each other and how that in turn affects the migration behavior of the tumor cells, because that can shed some light in the metastatic behavior. So we think that the system can be very useful for personalized medicine. For instance, the tumor biopsies can be isolated from the patients, and that those cells can be grown in our paper-based platform. Then those cells can be exposed to different types and different doses of chemotherapeutic agents, right? As well as different doses of radiations. And in the end, whichever therapy those cells are responding the best can be selected specifically for that particular patient. Maybe some of the advantages of the paper as a material, right, as a substrate, why is it good to use as a cell culture substrate, right? Because it is low cost and widely available material. It's available in many different chemical functionalities, different pore sizes, different thicknesses. It's very flexible. It's because it's an old material, it's very well characterized materials. It allows us to use multi-layered scaffolds, and it allows us to generate, because of its flexibility, it allows us to generate complex three-dimensional architectures. It also allows us to do high throughput sample preparation and analysis. And it can be a very useful platform. We can study the behavior of different cells from organs, and we can use that as like an organ on paper model, similar to lab on a chip applications, right? So there's been examples for microfluidic platforms being used to study the cells in different organs, right? Those are like organ on chip type of applications. And here we can, we can apply the same principle, and we can make organ on paper models. For instance, the multi-layered structure that I mentioned, we could easily include different materials and different cell types and study how the cells from different organs are interacting with each other in our organ on paper platform. So it can be very useful not only for repair and regeneration of tissues, but also using it as a disease model, using it as a personalized medicine platform to study and understand diseases also. It will be very useful. Absolutely. And one other research expertise that still the last thing that I wanted to mention, one of the other research expertise that we have in our lab is the point of care diagnostics. And here we also use paper to develop biosensors for detection of diseases. That includes viral and bacterial diseases. It includes tropical diseases, different health conditions such as allergies. We've looked at the presence of different cancer antigens. We've used those biosensors for forensic applications. We've detected the presence of illicit drug use, and we use them to test environmental agents for food and water quality. And more recently, we developed a rapid diagnostic test that can detect the COVID-19 virus. Earlier this year, when the public services reopened, right, it became very clear that there is a tremendous need for individuals to be, for some of the individuals, right, to be frequently tested and monitored for the COVID-19, which brings a significant economical burden. So especially frontline workers, service members, law enforcement officers, students at schools, and also some of the other members of the public, they need to be regularly tested. They need to perform routine tests, right? And the main detection methods for the COVID-19 include nucleic acid detection, real-time polymerase chain reaction, also known as the PCR test, also high throughput sequencing from nasal swabs or blood. But a lot of those techniques are time consuming. They can take from hours to days to perform. They're expensive and they require highly trained personnel to execute the test, as well as the requirement of specialized instrumentation, sophisticated instrumentation. So, and because there is insufficient number of diagnostic tests, sometimes only the patients with critical symptoms are being tested. And to address those limitations with the currently available methods in my lab, earlier this March, around March, April time, we developed a rapid and low cost, easy to use point of care diagnostic device that is portable and that can detect the COVID-19 virus in only 5 minutes using bodily fluids. And we can detect it at a wide range of concentrations. So the main advantages of our test is it's very rapid, it's portable, handheld, lightweight, and simple and affordable. It can be conveniently used at homes as well as in clinical facilities. It can be used by first responders or in decentralized care centers, as well as remote locations. We can utilize different bodily fluids to test the samples. For instance, we can test blood, urine, saliva, nasal fluids. In principle, the test is similar to home pregnancy tests, so even untrained users are able to execute the test and easily evaluate the results, easily read the results if the virus is present or not in the sample. And the devices that we make they're not sensitive to environmental conditions. So far, we haven't seen other products to reach the speed of our test. At this point, we need to test patient samples in our devices, and we're open for collaborations also for commercialization and translation of this product.
Wonderful, wonderful, amazing range of things you have going on. Yeah. I just heard about the point of care work that you were doing on top of all this, but that's also a fascinating connection to the underlying platforms. I just want to ask you one other question before I sort of get to the sort of the wrap up, and that's you've had obviously a lot of experience in the cardiac space, which is obviously always a hot topic, not just because it's still it still battles with cancer as the #1 killer of us all, but sort of this issue that the regenerative medicine, the engineering folks have struggled with in terms of vascularization, and you've written a lot about sort of oxygenation and so forth. Any interesting stuff going on in the lab with specifically regarding cardiovascular potential applications, whether they're now or looking out in the coming years?
Currently, we do have ongoing projects to study cardiovascular regeneration, how we can regenerate the damaged heart tissue, for instance, after a heart attack. And with a lot of the currently available materials, they're typically diffusion limited, but because if you're talking about a clinically relevant size construct that is typically in centimeter scale, and there's usually diffusion limitations to oxygen. And we are developing materials with biomaterials with high oxygen content that they can generate and release oxygen in a sustained manner for extended periods of time, up to 5 to 6 weeks. Currently, we're working on those projects. And it will be really exciting to share the results. We got really exciting results, and it'll be really nice to share them. We're currently writing up manuscripts, and I'll be able to tell more about those probably soon.
Outstanding, outstanding. Coming back to you for a bit of the wrap up, we typically ask our guests. Needless to say, you've no doubt met many fascinating folks along the way in academia and throughout your career. Any specific thought leaders, mentors, teachers throughout the path so far for you that have really been very instrumental that you may want to highlight and shout out to at this point, that, if it wasn't for them, Dr. Gulden Camci-Unal would be doing something entirely different with her career?
My PhD advisor, Professor Nikki Pohl, and my postdoc advisor, Professor Alika Damascene, had important influence over my career. Because of the flexibility that my PhD advisor provided me, I was able to expose myself to different aspects of my projects and develop skills to think outside of conventional approaches. And that's something that I continued doing over my career. And with my postdoc advisor, I had the opportunity to get involved in multidisciplinary projects in different research areas, and that helped broadening my perspective, and I continue to get involved in new research directions over time.
Inspiring stuff. Really wishing you the best with it. You have a lot on your plate, and in addition to the martial arts, quite a bit going on, and really want to thank you for taking the time. For everybody that's going to be listening to this episode across the podcast networks or watching on the YouTube channel, you've been listening to the amazing Dr. Gulden Camci-Unal, assistant professor in the Department of Chemical Engineering at University of Massachusetts Lowell. Dr. Unal, thank you for taking the time to come talk with us today and sharing your work, your visions, and everything you're doing. And as we say on the show, thank you for moving the human story forward. It's really inspiring stuff, and I take my hat off for for everything that you have on your plate. But really, really eye-opening. And thank you for moving the future a bit closer with what you do.